Okay, I hope everything is fine here now. Students, please comment and let me know you are in the in this thing or not. So, hello. So basically, what I need to do is I understood. Okay, so yeah, okay, fine. I'll just wait for some. So yeah, basically this is the no, this is not the old way, Agrudeep. This is the new way only, but I have just done some kind of joratali here. So let's see how much it works. Maybe it is not being able to take the pressure. I think little bit of engineering will help here. So okay, so fine. So let us go into the discussion. So biotechnology, we were discussing about RDT, right? So there is a target gene, and that target gene has to be cloned. So what are the basic steps is that we have to first okay. So what is the basic thing that we have to extract the target gene? First thing that comes to our mind. Second is we have to amplify it, amplification of the target gene. We have to amplification of the target gene. So that is what we will do. Second, and then third is extract the DNA, extract the DNA or the product. Now as you know, it is this thing, if I want to diagrammatically represent, then how it would look is it would look like this. Suppose there is a gene segment, okay, there is a genetic material. Suppose this is the interest of gene at this point to this point. This is the target gene, okay, this is the target gene. Let us assume that this is the genomic segment from the beta Langerhans cells of pancreas, okay, and pancreas, and hence this target gene is of, we could say that this target gene is of maybe insulin, okay. So this is what it is. Now what you have to do when we separate out or cut out the target gene, then I had said you previously that to cut the target gene, to cut open the target gene and break it out, we need the special quality of enzymes. Those enzymes are known as restriction endonucleases. Okay, endonucleases. The term endonuclease breaks down or cuts nucleic acids, nucleic acids. Those are called nucleases. And when we use the term endonucleases, means breaks down or cuts the nucleus from inside the sequence, so that is known as a endonuclease. Then why is it called restriction endonuclease? Because it first of all it cuts as a specific site. And why is it called restriction? We will come into it in a few minutes. Okay, I will come into this discussion that why it is called restriction endonuclease after a few minutes. But after apart from that, we will try to understand that after using the restriction endonuclease, we cut here the enzymes. They are also known as molecular scissors. Molecular scissor. Okay, so this molecular scissors or restriction endonucleases will be used to cut the target gene. And then what will happen when the target gene is already outside? Suppose this is the target gene which is already outside. So this is your target gene. Okay, now the target gene is already outside. Now what you have to do, you cannot take this target gene and directly put it inside a bacterial cell. Suppose you want to take the target gene and put it inside a bacterial cell, and you want this gene to be expressed or to be multiplied or to be cloned. Okay, two things you can aim. See, whenever you have a target gene, you can aim two things with it. You can aim two things with it. You can aim two things with it. What are the two things that you can aim with it? The two things that you can aim with it, one is you can aim for its expression. If you aim for its expression, means you are interested in the product. Okay, and another thing, you can just aim its cloning, means just increasing its copy number. Okay, so this is what you can do. This is basically what is, what are the two things you can do with the target gene. Now suppose, for example, either for expression of the product inside the a cell which you are using, where you want to amplify this gene, or we are talking about amplification. First you have extract. I have talked about extraction. First you have extracted. I have talked about extraction. That extraction, how you can do is you can take it out with the help of restriction endonuclease. However, only restriction endonuclease will be required and others will not be required. That is also not true. But however, you see, what happens is that once you take it out and once you get the target gene, what you can do is you can put it inside the cell. But do you know that if any, do you know this thing, that if any, here one thing I'm bringing and saying that from here to here I'm bringing it down, or I can say it over here also, that if any foreign DNA enters E. coli cell, suppose for example E. coli cell, this is, I'm talking about E. coli cell, but I will also this is also true for other cells. If any foreign DNA enters inside the E. coli cell, the E. coli cell has mechanism to break it down. Because this is like we have a defense mechanism. All the E. coli cells or bacterial cells, which are basically the host cells, for what we can say, for amplification of the products, or for taking it, so not an amplification of the products, taking the gene in so that they can be expressed or being multiplied. Suppose for example, if there is any host cell and in which any foreign DNA is entering, suppose for example any foreign DNA is entering, it is not that easy. Because why I am saying so, it is not that easy, because suppose for example this is a cell, bacterial cell or E. coli cell. Okay, if simply a virus comes and attacks, what is the mode of or mechanism of infection of virus? Suppose this is the cell. What is the mechanism of infection of virus? Virus introduces the DNA inside. Okay, when the virus introduces the DNA inside, now when we find the DNA or the RNA, whichever inside, the E. coli cell itself has a defense mechanism which is called a 2-complex system. Complex system. In 1963 it was first discovered, and from there only, this discovery was very important for our biotechnological perspective, because this 2-complex system involves two things. One is methylation One is methylation and second is endonuclease. Actually, the fact is that what will happen is the E. coli cell, the system itself has an endonuclease which will go and cut the foreign DNA. Cut the foreign DNA. Okay. Now, when the endonuclease will cut the foreign DNA will automatically get degraded and E. coli is already saved from the virus. This is a defense mechanism. Okay. And then, sir, what is the importance of methylation? Now, I'll speak about what is the importance of methylation in a little bit, Let's see. What happens is, suppose this is the bacterial DNA, bacterial cell. Okay, suppose the bacterial chromosome is there. Okay, this is the bacterial chromosome, and a virus has come and has injected a foreign DNA. As for that matters, not only the virus will inject the foreign DNA, but maybe you are injecting the foreign DNA or trying to make it transform. Transformation is a different thing that I will talk. Okay, but a foreign DNA, when it tries to enter, then what happens? When the foreign DNA tries to enter, then the two complex system works. Two complex system starts working. Now, when the two complex system starts working, you will think that, okay, fine, that in two complex system that I am talking about, there is what we can say, there is DNA methylation and endonuclease, like sir has mentioned, and fine, one part of the system, endonuclease, will go and break it down. Okay, but the fact is this: if this endonuclease can break down this DNA, suppose a endonuclease is there and a DNA strand is there, and between A and T it goes and cuts the DNA. Cuts the DNA means breaks the phosphodiester bond. Breaks the phosphodiester bond, because we already know that A और T के बीच में यहाँ पे एक ही strand में the phosphodiester bond is there. It may be A or G also, it may be C and T also, whatever it is, but in a sequence, whenever in one strand we are talking about it, there are phosphodiester bonds, and if there is another strand, then there are hydrogen bonds that you know. Hydrogen bonds are A double bonded to 2 and G triple bonded to C. This is different. Okay, here we are talking about the phosphodiester bonds. Now what will happen? The endonuclease will go and cut it here and करके खुल जाएगा. But the fact is this: why do you think that this endonuclease will break down this foreign DNA and not break down? Do you not think that A and T are also present in the bacterial chromosome? So the endonuclease would be already breaking down the bacterial chromosome. When the endonuclease will already be breaking down the bacterial chromosome, then that will be a different case. The bacteria would be able to survive. So how is it taking it? It would be like a self-suicide. Suicide, isn't it? So would the bacteria prefer to do that? No, definitely no. The bacteria wouldn't prefer to do that. As I am trying to say, the bacteria wouldn't prefer to do that. So what would I like to say is that this is being avoided by the process of methylation. How this is avoided? The process of methylation is that what it does is it adds methyl group to the A and T in the sites where the endonuclease. This is the reason I told you. I will tell you that why it is called endonuclease. I explained, but why is it called restriction endonuclease? I didn't explain. Now I will try to explain why is it called restriction endonuclease. You see, the fact is this. Oh my God! Wait. Wow. You see, the fact is this, that it is called restriction endonuclease because this endonuclease is restricting the foreign DNA to take over, to infect the cell. That's why it is called restriction endonuclease. Now, restriction also means restriction endonuclease. It also means restricting, and also it has specific site of cut. Okay, so this specific site is cutting here, and here the specific site of the specific nucleotides that is available, that is methylated, because this restriction endonuclease is an enzyme, is a specific structure which would already be able to break the phosphodiester bond between A and T and not between methylated A and T, because it will not recognize as A. The restriction endonuclease will not recognize this A as A because it is methylated. So this is the importance of methylation present. So this is two complex system, defense mechanism of E. coli. Now the fact is this, that this was discovered in 1963, and why is this discovery important? Why is this was discovered in 1963, and why is this discovery important? That is the fact. Why is this discovery important? Because from this discovery only we came to know about endonuclease, and today endonuclease is so important that we create cuts through endonuclease in various sites of human genome even, or plant genome even, and take out the target DNA and use it for amplification. But the problem is that this foreign DNA could be also degraded by the host, what system? The defense system, which is this two complex system. That happens in case of a viral infection, or the virus which infects a bacteria. What it is called? It is called a bacteriophage. The virus which infects a bacteria is called a bacteriophage. Always remember, a virus which infects the bacteria is called a bacteriophage. Now the fact is this, that okay, this happens. So this is the first step that we have understood, that okay, discovery of endonuclease was also from here, and this endonuclease is only we are using for cutting down and again taking up the target DNA and taking the target DNA and putting it inside the host cell. There should be method. You cannot put the naked DNA inside the host cell, because first of all it has the chance of getting degraded, and second thing is that when it has chance of any thing, it will absorb and it will cut, and second thing is that that if it only a naked or simple DNA strand enters inside the cell, it doesn't have a origin of replication, so it cannot multiply inside, or it cannot be before cell division a particular copy of it should be made. So replication of this target gene should be there. So origin of replication is not there, simple segment. So what is not there? Origin of what is the problems? The problems are, if this target DNA is simply inserted into the bacterial cell, where is the origin of replication? Where is origin of replication? So first thing, or the first problem that is there, is that the origin of replication is not there. Okay, so who is this Shed Music Ray? This is bio of class 12 CBSE anyway. Who is this? Wasn't the thing unlisted? It's okay, but who is this person? But in the unlisted video, how can a unknown person come? My video is unlisted. Let me check. That means someone is sharing my thing, whenever I am sharing it. Anyway, to answer the person's question, this bio of, I have checked it is unlisted. It is always unlisted. Okay, anyway, don't use derogatory words or something like that. Yeah, we should continue. Anyway, so the target gene do not have a origin of replication. When it doesn't have a origin of replication, we cannot expect that when this cell will divide into two, then this along with the chromosome will come in both the thing, because you know that DNA replication is very important, so that the both the cell receives the gene. And normally if you see DNA replication, if you see this, normally if you see, and normally it is replicated, then what happens? The replication of this will occur, two copies will occur, one copy will come in one cell, another copy will another cell. So if there is a target gene and it doesn't have an origin of replication, so it will come in only one and not come. So what is your I don't know how to block. Anyway, so what is your what to say? What is your agenda of putting this? You wanted to amplify this target gene. You wanted to amplify. So is it really getting, oh my god. So is it really getting amplified? No, it is not getting amplified. It is not getting amplified at all. So that is the thing that I wanted to say. So definitely simply entering the target gene would create two problems. First is that it has big chance of getting degraded due to the two complex system. Second is not having origin, it won't be amplified, it won't be replicated, it won't be replicated. Not having the origin, it won't be not having the origin, it won't be replicated, or what to say, or amplified. No, origin of what? Origin of replication or amplified. This is the first, second thing. And third thing is that not having, does the single gene segment have any start codon or stop codon, maybe the promoter, so not any promoter or regulator sequences. Or regulated sequences. So these are the three problems that will be there, which won't allow us that, okay, we will put just the target DNA just after taking the, whatever we say, that after just taking the target DNA, we will take it and insert it. No, that cannot be done. So that is the basic thing that I wanted to say you. That is the first thing that you have to understand. Okay, so what we need, so first thing we understood, what we need is restriction endonuclease. This is the one big thing that we need, restriction endonuclease. And the second big thing that we need is a vector. What is a vector? Vector is a vehicle, is a molecular vehicle. Okay, so the best vector that we can talk about is, vector is a molecular vehicle, and the best vector that we can talk about is plasmid. There are a lot of vectors, plasmids, cosmids, bacteriophages are also vector. You know, inserting, it is also a technique. Suppose a bacteriophage, that is the bacteria infecting virus is there. Inside it, you put your target genome and, you know, allow this to infect E. coli cells, and that can also happen. So it will insert the genome, because it is the mode of infection by, is the mode of infection, I'm sorry. না না না, তোদের কিছু করার দরকার নেই, ঠিক আছে থাক, দেখি না, পরে বুঝতে পারবো। আনলিস্টেড তোদের মধ্যে থেকে কেউ শেয়ার না করলে তো হবে না এটা। যাই হোক, তোরা ফোকাস কর যেটা আমি করাচ্ছি সেটাতে, এটাই বেস্ট হবে আমাদের জন্য এখন, ঠিক আছে? তো দ্য প্লাজমিডস আর দ্য মলিকুলার ভেহিকেল, ঠিক আছে? বা অনেক সময় দ্য মলিকুলার ভেহিকেল অর দ্য ভেক্টর ক্যান বি ব্যাকটেরিওফাজেস অলসো। There are various types of vectors. We will learn in detail about the vectors. That is different thing. Now, when we talk about plasmids, see, plasmids are what? Plasmids are self-replicating circular DNA molecules. Plasmids are self-replicating circular DNA molecules. ঠিক আছে? ধর একটা ব্যাকটেরিয়াল সেল আছে, ঠিক আছে? এখানে ব্যাকটেরিয়াল ক্রোমোজোমটা আছে, দিস ইজ ব্যাকটেরিয়াল ক্রোমোজোম, or the main DNA, bacterial DNA. Apart from this bacterial chromosome, they have small, maybe one or maybe multiple copies. What did I say? Maybe one or maybe multiple copies of small circular DNAs. Okay, that are there. Now question arises that do the bacteria naturally carry them? Yes, bacteria carry, some bacteria carries them naturally ক্যারি করে। তার মানে কি করে, যখন ধর একটা ব্যাকটেরিয়াল সেল আছে, when there is a bacterial cell, suppose a bacteria, and it has suppose plasmid DNA, then when it will divide into two, okay, when it will divide into two, the bacterial chromosome will also be replicated and the plasmid will also be replicated and made two and transferred into two. So replication of both the bacterial chromosome and the plasmid DNA occurs. So this is the thing that you should keep in your mind. So lot of basics that I have told. Now these plasmids can act as vectors. How I am telling you this, as because this, now see, these plasmids are replicated, that means definitely, this plasmids, replication of these plasmids occur. So that means definitely the plasmids have origin of replication. Okay, so I'm sorry. Anyway, so that means definitely the plasmids have origin of replication. Okay, so if the plasmids have origin of replication, so the plasmids can replicate. Now our what we can say is that our thing that we can do is, we take the target DNA, this is a bacterial cell, okay, and when we take the bacterial cell, we take the target DNA, fine. Okay, we take the target DNA and what we take is, we take the plasmid. Now we cut the plasmid, with restriction endonucleases, REs. Okay, and this gene segment has been cut from a gene, suppose a chromosome, this gene segment has also been cut, okay, with the same RE. Now why is it important? Now question arises, why is it important to cut with the same restriction endonuclease? Why is it important to cut with the same? It is important to cut with the same restriction endonuclease because we want to create same type of cut ends so that the target DNA and the vector gets ligated easily. Ligated means joda laga. Okay, which enzyme does the work of this ligation? Ligase. Ligase are known as molecular glues. ligase are known as molecular glues. They are known as molecular glues. So that is the thing that you should understand. And now what we will do is we will cut this, and when we cut this, this is created, and what will happen is this will come and join here. Okay, so this is our rDNA. We call it rDNA or recombinant DNA. We called it rDNA or recombinant DNA. Now tell me, up to this much, is this the thing that you have understood? Please comment and say. Please comment and let me know that have you understood this? I don't know how many of you are online. Okay, good. Good, you are listening and understanding. Do you know the first rDNA? Do you know the first rDNA? If I ask you which is the first rDNA, then the first rDNA was discovered in 1972 by Boyer and Cohen. Okay, the first rDNA was discovered. What the nonsense is going on? Anyway, so yeah, in 1972, I'm sorry, there were some disturbances outside. So anyway, in 1972, Boyer and Cohen, these were the two people who discovered the rDNA for the first time, or the recombinant DNA. rDNA means what? Recombinant DNA for the first time. And this was done by as you know, in Salmonella, the bacteria Salmonella typhimurium. Salmonella typhimurium, in the bacteria Salmonella typhimurium, where an antibiotic resistance gene was incorporated in the plasmid of Salmonella typhimurium to create a recombinant DNA. So that is what is the first rDNA ever. Okay, so I hope that you have understood up to this much and everything is clear with you. Now moving to okay, first we have understood that which is the first rDNA. We have understood everything. Now we have also understood that what should be done and what not should be done, and from where the discovery of restriction in the nucleus is there, and why do you have to put it inside a plasmid. Because when you put it inside a plasmid, here the question that I have asked, that yes, you cannot put, here the reasons that you cannot put the target symbol, the target gene inside this, because there are a lot of problems that I have talked about here. And then I also would say that fine, if the plasmid itself has a ORIC, the plasmid itself has a ORIC, or ORIC means origin of replication, then if you insert the gene in a plasmid, naturally what will happen, when the plasmid is inside the cell, it will, suppose this is your gene of interest that has been there, and this is the bacterial chromosome. Now when it will divide, the plasmid having the origin of replication, it will replicate, and hence your target DNA will also get replicated and come into each of the daughter cells. Right or wrong? And come into each of the daughter cells along with the host chromosome. So I hope that you have understood this particular thing, that what I tried to mean, that why you put inside a plasmid. Okay, but here the importance of putting inside the plasmid doesn't end. So the second thing is that it will not get easily degraded by the endonuclease, because it is not a naked DNA. The plasmid is already inherently carried by the bacteria. Now the fact is this: there is a natural process that you should know. There are three natural processes. Now I hope that up to this much you have understood everything. Okay, this is the basic understanding of molecular biology. Now what happens is, you see, there are three types of, now keep these informations there that are that I have given you, this all informations that I have given you from today. Okay, this, first of all, this, okay, what is biotechnology, what is recombinant DNA technology, what happens in recombinant DNA technology, what is a recombinant DNA, what are restriction endonucleases, then what are endonucleases, why are they called restriction endonucleases, why are they called restriction, why are they called endonucleases, when it was discovered, and what is two complex system, what is methylation, importance of methylation endonucleases, and what are bacteriophages, and what will happen to a simple DNA or target DNA if inserted naked inside the bacterial cell, or any cell as for that matters, and what will be the problems that will be created, which we have discussed, and we have also discussed that what are plasmids and what are their advantages and where they are present. But do you think why bacteria carries plasmids? Why bacteria carry plasmids? Bacteria carry plasmid naturally, but why bacteria carry plasmids? What is the requirement for bacteria to carry plasmid? Bacteria generally carry plasmid because apart from the normal genes that are there inside the chromosome for their amino acid biosynthesis, maybe respiration, glycolysis processes, of which it will undergo, okay, which it will undergo, like I have said. So that is there, but also it will carry some extra genes for extra characters, like one of them is antibiotic resistance. One of it is antibiotic resistance. You know what is antibiotic resistance? Antibiotic resistance is resistance to the antibiotic, because when bacteria grow, suppose when bacteria grow in a medium, okay, suppose naturally, what happens, many other bacteria also grow. Suppose some other bacteria are also growing, some other bacteria also, and they release some certain toxic substances so that these blue bacteria die, because they want to win the competition and take all the food to their name. But what happens, some bacteria then develop those chemical which kills other bacteria, is known as antibiotics. Okay, now some bacteria has developed resistance to it, and they are called antibiotic resistance. Because even if they, okay, fine, you are eliminating out of the competition, fine, I will develop resistance to my to your chemical. So that type of genes of developing antibiotic resistance is carried in plasmid. Also some other characteristics also there. Maybe suppose there is a bacteria called, I'll talk about it. There is a bacteria, Agrobacterium tumefaciens. Agrodip, it is you. Tumefaciens. It causes tumor to plants. Okay. Now this bacteria carry a plasmid called TI plasmid. Now this Agrobacterium tumefaciens causes tumor to plant, and those disease-causing genes are present in the TI plasmid. Okay, remember this. So those disease causing genes are present in the TI plasmid. So here we can see that some extra, what we can say, characteristics like pathogenicity. So one is antibiotic resistance. What is the other extra characteristics that it can carry? That is pathogenicity. Number two, genicity that it can carry. So these are the things that are carried by bacteria inside a plasmid, apart from its normal genes of normal, yes, crown gall disease. You are not a crown gall disease, but you are Agrobacterium tumefaciens, which causes the crown gall disease or tumor in plants. So this is the thing. Now, I have not told you the disease. I have told you the bacteria which causes the disease, and you carry the. It was just a joke though, because your name matches, no, Agrobacterium, Agrodeep Agrobacterium. So that's why. Anyway, so that is the thing I wanted to say you. So yeah, so the pathogenicity and these things are there in the plasmid. So this is the reason why bacteria carry plasmids. So bacteria may carry plasmid because of this. But however, if you have understood this entire thing, and those plasmids are only used as molecular vectors. Now only plasmids are used as molecular vectors. No, bacteriophages may also be used. Bacteriophages may also be used as vectors. That also you should keep inside your mind. There are cosmids, lots of vectors. We will discuss vectors in details at that class. We will discuss just tomorrow only. We will do the class. Now there are three types of natural processes. Natural processes of bacteria which you should know to understand the process of biotechnology properly. Number one is transformation. Number two is transduction. And number three is conjugation. Now, when I will tell about these processes, you will know that why these processes are so important, and I'm saying why I'm talking about these processes. Okay. So, first of all, when I talk about transformation, suppose a bacterial cell is there and a naked DNA or a naked plasmid is roaming around. So, through the pores or through the pores of the cell wall, the plasmid or the DNA can enter, or it takes up DNA molecule or gene segment. DNA molecule here means plasmid only I am talking about. Okay, segment from environment. So this is known as transformation. The process of transformation can be enhanced. You know how much transformable a bacteria is. Suppose you have put a medium where bacteria can grow, and you have put naked DNA inside it, and you have put bacterial cells also. So how much it can enter, that the amount of the capacity or the capability of bacteria to take up the DNA is known as competence. Okay, and the one which can take up is known as a competent cell, and the one which cannot is known as a non-competent cell. Okay, so that happens, and you can increase the competence. I'm so sorry. Okay, you can increase the competence by adding CaCl2. This increases the pore size actually of the bacterial membrane. That's why the competence increases, or increases the numbers of the pores. Transduction is, suppose a bacterial cell is there and a phage comes, a bacteriophage, a bacterial virus comes and injects its phage's genetic material inside the bacterial cell. Or what you can do is, you can load a phage with the help of you can load a phage with your desired DNA and allow the phage to infect the, or bacteriophage to infect the bacterial cell. This process is known as transduction. So one is transformation, one is transduction. So I hope that you are understanding that these are the processes to transfer or use the vector to transfer the target gene inside your cell. So that is the thing. I hope you understand that there is the natural process, of bacteriophages. Oh my god. Okay. The natural process of bacteriophages to infect bacteria is that what it will do is the bacterial cell is there and the bacteriophage will come. And what is a bacteriophage? A bacteriophage is a virus. Normally viruses are how? Viruses have a protein capsid. I'm sorry. Okay, so normally viruses have a protein capsid. Okay, so inside the protein capsid there is DNA. This DNA codes for the proteins that makes up the capsid. Capsid means the outer covering. So the protein capsid makes the outer covering, and inside the DNA of the virus is there. This DNA carries the sequence for making this capsid. When this DNA is injected in and is using the bacterial host machinery enzymes to multiply copies this DNA and express them, then what will happen is again the new viruses will be made, and these viruses will lyse the cell, and these viruses will come out, okay, and infect new bacteria. Okay, so this is the infection cycle, or this is also known as lytic cycle. Okay, there is also another cycle of bacteriophage that I'll just tell. This is known as the lytic cycle, where the phage viruses brings its DNA. In lytic cycle, what is happening is the phage viruses, it's bringing its DNA and making it multiply, and using the host machinery expressing the DNA or doing the central dogma of molecular biology in the host cell of its own DNA, and creating multiple copies of the virus okay, and infecting the cells, okay, and hence increasing the number. Okay, so that is lytic cycle. There is another cycle known as lysogenic cycle. In lysogenic cycle, what happens? The phage virus comes, or the virus comes, and whether it will do lytic cycle or lysogenic cycle, that depends on the type of the virus. Remember, when it is, I'm not writing everything, but I'm giving a lot of information, so you should remember whether it is going on lytic cycle or it is going to undertake a lysogenic cycle. It depends on the environment and also the type of the phage virus. And when it will happen, what will happen is that the DNA will come down, and the DNA will itself insert in the bacterial chromosome like this. So, and it will stay there, and the bacteria will multiply. Then then what will happen? Continuously the DNA will also be multiplied. The bacterial phage DNA is also getting multiplied. Later on, when the environment becomes favorable, then it goes or shifts into lytic cycle and then bursts the cells and comes out. So this is what it is. But the process of transfer of this, using this as a, when we study or know this natural process and exploit this natural process to transfer our own desired gene into this, we call it transduction. And then there is conjugation. Conjugation is nothing. Suppose one bacteria contains one plasmid apart from its chromosome. This is the chromosome, suppose. It has a pili. It is known as a F plus bacteria because it has the plasmid. And then another is there, it will join, both will join the pili. It doesn't have the plasmid, but it has a multiple copy of the plasmid. One plasmid will be transferred here. In this way also, you can naturally the plasmids gets transferred and increases in number, and the process is known as conjugation. Now tell me, did you understand everything? Whatever I have talked? Okay.